Hey guys, today we're going to be talking about our mannitol salt auger plate, or what we simply call the MSA plate. The MSA plate is a differential and selected medium generally used for the isolation and classification of staphylococcal species. The MSA plate has mannitol in it as its only carbohydrate source, which is sort of its differential component, and a 7.5% sodium chloride concentration, which is its selective component. It also has the pH indicator phenol red in it, which will be yellow at around pH 6.6 and below. Between pH 6.6 and 8, it will have varying shades of red, and then around pH 8 and above, it's going to be more of a purple color. So with that in mind, let's talk a bit about our media basics, and we'll start out with mannitol fermentation. Basically, any organism that is able to ferment mannitol will create acidic byproducts, which lowers the pH of the medium and changes it from this kind of reddish color to more of a yellow color like that. Now, a thing to keep in mind is that the medium will also have some protein in it, and if those proteins are broken down through deamination reactions, that will produce alkaline products, which increases the pH of the medium, turning it more of this sort of pinkish purple color. On the selected component of our medium, again, it has that 7.5% sodium chloride concentration, and that creates a very salty environment for most organisms. Basically, most bacteria, other than the staphylococcal species, won't be able to survive or grow in that type of environment. So generally, we can say that if you see growth on your plate, it's most likely going to be a staphylococcal species. So kind of with that in mind, let's go ahead and talk results. So after you've inoculated your plate and you're coming back after the incubation period, what can you see and how can you score that? So regardless of the color of your plate, as long as you're seeing bacterial growth on the plate, you can assume that that is going to be a staphylococcal species. So again, that growth can look kind of like that. It might look more like the colonies that you see on this plate, a little bit more spread out like that, or thicker colonies. But again, regardless of the color, if it is yellow, red, or purple, if there's growth on the plate, you can say that it is positive for your staphylococcal species. Now let's go ahead and talk results in terms of our mannitol fermentation. If your plate is yellow after incubation, that is going to be a positive result for mannitol fermentation. If your plate is red after incubation, that is going to be a negative result for mannitol fermentation. And if your plate is kind of this purple color after incubation, that is also going to be a negative result for fermentation. So deamination reactions tend to occur after long incubation periods, but with the mannitol fermentation, normally if that does occur, it will create so many acidic byproducts that your plate should not change from the yellow color back to a purple color. It should stay yellow for a very, very long time. With that out of the way, let's go ahead and talk how we inoculate our plate. So in terms of inoculation, you can go ahead and just do a standard broth to plate inoculation. That's perfectly fine. If you have your sample on a slant or on a plate, you can do a standard slant to plate or plate to plate inoculation as well. Now, the last thing we do have to talk about with the MSA plate, which is just a little bit more of a side note, is that since it does have such a high concentration of salt in it, it can dry out very quickly. So with that in mind, sometimes it turns into kind of like a little potato chip is what it looks like. The media gets really, really thin, or it might start crystallizing and you get almost this sort of lightning pattern of crystals on top of the plate. Those patterns are not growth. So if you see again this very scaly or dry looking plate, it's just that it has dried out. The easiest way to try and prevent our plates from drying out is actually just by incubating them at 25 degrees Celsius instead of 37 degrees Celsius. The 37 degrees Celsius will drive out a bit more water out of the plate quicker, so leaving it at 25 degrees will just be better overall for maintaining the plate for a longer amount of time. But that should cover the items that we need to know for our mannitol salt auger